Our project is a practical method of categorizing poisonous mushrooms and species diversity. We have three hypotheses we hope to explore. Our first hypothesis is that mushroom features can be used to predict whether or not they're toxic. Our second hypothesis is that mushroom's habitat affects whether or not the mushroom is toxic. And the third hypothesis is that level of species diversity affects whether or not a mushroom will be toxic. We have data of over 8,000 different mushroom samples, and they're listed these different physical features, and we have this correlation heat map of all these different features. So we have created different classification models, classifying them to be toxic or not. And our favorite, of course, is this decision tree because it's visual and we can see exactly what's going on here. And it's awesome because it has 100% accuracy on test data, which supports our first hypothesis. Here we have our second hypothesis test, and we have divided the mushrooms by habitat and performed a z-test on each one. And we have found that yes, each habitat does affect whether or not the mushrooms will be toxic.
The second part will explain about the correlation between species diversity and mushroom development. Here we have phylogeny tree of species from our data. This tree explains about the division of habitat, which is wood, grass, and waste. Based on the division, we made diversity map. The diversity map showing the concentrations of species in each habitat coordinate. Red dots are showing the all species, and blue dots showing the waste, and green dots are showing wood habitat, and the purple is grass habitat. Since the overlapped dots couldn't display it on the map, the frequency and diversity of species in each coordinate were organized into data frame.
Finally, we have our third hypothesis. So we have divided the mushroom habitats into groups. The first group is habitats with a dense concentration of species diversity, and our second group is habitats with scarce concentration of species. We performed this two population proportion statistical inference test using the mushroom data, the percentage of in each habitat of toxic mushrooms, and we have found that yes, mushrooms in habitats with higher species diversity are more likely to be toxic. So that supports our third hypothesis.
Conclusion: we were able to support our hypothesis by our data. We believe that we can proceed future research by broad data and method.
So that's what we have. Thank you.